Hello, everybody. Hi. I don't know if is this mic on? You're all good? Thank you. It did work, much better than me yelling. Hi, everyone. Thank you so much for coming to Science Disrupt London Sessions, Disrupting the Lab. This is actually our biggest audience, so thank you to all of you for coming and joining us. My name's Gemma. This is Lawrence. We're co-founders of Science Disrupt. We've been going since April. We started off as a podcast, interviewing interesting innovators, startups, researchers, anyone who I guess we were thinking is changing the way we do science. And we now obviously have moved into events because we want all of you guys to meet each other, collaborate, chat, have drinks, have mince pies, all that sort of thing. I wrote down my notes, but I totally forgot to bring them out to tell you. So, okay, so who we are, that's cool. Oh, we've also got Ignacio, where is he? He's here. So he's also helping out tonight as well for Science Disrupt. So, things I want to mention, in case you're wondering about our podcast, which is our main content, our most recent episode was with Shafi Ahmed and Steve Dan, and they're talking about changing the way we do surgery using VR. It's a really cool episode, so please check out if you're remotely interested in the future of health. And the next one we've got going is with Alice Bentink, who's the co-founder of Entrepreneur First. So if anyone's got any ideas for startups or whatever, they are looking for scientists, PhDs, postdocs, whatever. So please listen in and get involved. We also have a Slack. For anyone who doesn't know what Slack is, it's kind of like a cross between WhatsApp and Facebook and email. It's basically an online community for lots of people to come chat. We have one specifically for science disruptors. Please join. There's lots of channels on there to kind of find new people, find new ideas, generally talk about science and doing things differently. We'll send out the link to that in the next newsletter, which will come out next week, which will make sure you guys are all on. Opt out if you'd like to, but we'll put you on so that the Slack link is on there. Finally, I think Wi-Fi is here, and the hashtag. We made a really beautiful sign for you. Hope you like our design. So yeah, please feel free to tweet or whatever if you're into that. Finally, our structure tonight, so we're going to have 3 awesome speakers. Before that, we're going to have a little sponsor message. We're going to have 3 awesome speakers talking for 15 minutes about how they're disrupting the lab, and then we're going to have a panel at the end who's going to be moderated by Ali here, who's from HackScience. So please, if you wouldn't mind keeping your questions to the end for the panel, we'll try and get as much audience participation, and then afterwards we'll continue with drinks and you can chat to anyone. Please come say hello if you want, or not if you don't. And hopefully you'll enjoy the rest of the evening. But yeah, without further ado, Ali's gonna, oh no, sorry, not Ed's gonna start, apologies, all over the place, and tell us a little bit about Cloud of Experts, so thank you.
Thank you for that, Gemma, that's great. My name's Ed Cartwright, I'm the founder of Cloud of Experts, and I've decided to sponsor this event this month because I came a few months ago to the health event and it was great, and somebody bought my beer, so I thought I'd repay the favour as a small company and I'd buy everybody, help put towards money to buy everyone some drinks. So as a result of that, I want to take the opportunity to be a little bit nosy and find out a little bit about you and to segment the audience. I like doing this when I'm talking. If you can just look around, see what everybody else is, see who we have in the audience. So I'll do it first by education. So put your hand up, please, if you're still at university. Everyone put your hand up. Right, okay, that's great. So the rest of you haven't, that's great. Put your hand up if you have a bachelor's. Yeah, and keep it up if you've got a master's. Yeah, and then keep it up if you've got a PhD. Okay, wow, okay, that's quite interesting. I see all the other speakers kind of having a look around and seeing what the audience is, that's great. Do we have any medics here? Kind of one token medic, great. Okay. Do you have a PhD? Medic with a PhD? Okay, great, fantastic. Okay, cool. Come and speak to me later, great. Any computer scientists? Fantastic, great, cool. Right, anyone working for a startup? Interesting, okay, great. Anyone wanting to start a company? Yeah, fantastic. And anyone wanting to join a startup? Quite a few of you as well. Okay, great. And I doubt there's anybody here who's a funder, but I thought I should ask. Oh, okay. Okay. Hi. Okay. So a little bit about me. I'm talking for 5 minutes, guys. I'm not going to bore you. So my name's Ed Cartwright, and I'm a doctor. I qualified as a doctor about 10 years ago. I'm a paediatrician. I work frontline paediatrics, looking after preterm babies all the way up to very grumpy adolescents. It's great fun. It's a specialty that kind of isn't based on cardiology or kind of looking at the heart or looking at respiratory. It sort of has a lot of interest and value to it. I went and did a PhD, went to Cambridge and finished that about 2 years ago, and that was at the interface of microbiology, genomics, and epidemiology. During that time, I worked with industry. It was fascinating, eye-opening. What was really interesting for me was quite how much they didn't quite understand about my area, but that's not really that surprising. But actually, there's huge gaps about clinical knowledge that even really well-funded biotech companies just didn't know about. So I worked with them, and we had very productive collaboration. I went back to clinical pediatrics, and then what was very apparent to me was that there was a time lag between what was available in my lovely, shiny lab in Cambridge and what was available at frontline pediatrics on the bedside and near the bedside and in the microbiology lab. And the reason for setting up my company, Cloud of Experts, was to bridge that gap and to help it so that science comes to the bedside faster. So that's the basic aim. And it's a two-sided marketplace. And we specialize initially in providing medical doctors who have a PhD and linking them up with industry. And it fixes many of the inherent problems that occur between academic and industry knowledge exchange. And there are many, and I'm not going to go into them because that's not, this isn't a pitch, this is just a talk. And the message, the reason I was sort of trying to segment you is that I've got 3 messages. So I'm looking to some of the medtech founders who are in the audience who might be interested in what I do as a company. We have all medical specialties on our platform. So cardiologists, paediatricians, orthopedic surgeons, we've got a professor of plastic surgery from Oxford. And we link you with the experts who are extraordinarily busy. This is the way that we designed our site was so that we could minimize the amount of interaction that they need to do so that, from their end, they look at the job very quickly and decide yes or no and can kind of tell us in a very smooth manner whether or not they're interested. Medtech startups, but inherently you need to talk to doctors early on if you've kind of got a medical app, if you've got designing a medical device. It's really easy now to start a medical device in terms of kind of getting the initial design. Regulation is the big headache, and that's where kind of specialists in the field might be able to help. I can make it even cheaper for startups. I've had good success in this, in kind of just saying, look, this is a really small company. This isn't a hedge fund in Mayfair. This isn't kind of a big biotech company. Just help them out. Kind of just lower your rate a little bit for your hourly rate, and that's worked. I can help you as well, but with my own background with design, kind of putting questions out there for them. I'm running out of time a little bit, so I'm not going to go into work examples, but do get in touch if you think any of that is interesting. The second message that I wanted to give was to all medtech and life science tech founders, and briefly, there's kind of what are the comparisons between Silicon Valley and London? Silicon Valley is better at pretty much everything, right? Apart from maybe fintech, is that right? Is that kind of what kind of the popular press of kind of thinking? Is that there's really strength of London. There's fantastic companies that have been coming out, huge exits, lots of success. But actually, we are known for fintech. But actually, I think it should also be life sciences, and that's because of the strength of our universities that we have here, and it's because of the strength of the life science industry here. Selling to big tech, biotech, pharma, and medical device companies is really hard. It's really expensive to go to conferences. Let's talk about how we can help things out. So if you're another company, just come and let me know, get in touch. Finally, any software engineers, we're looking to expand. We've got traction. We're preparing to apply for funding. So if you'd like to get in touch, then do so. And this is the main slide with my contact details. On the Slack group that was mentioned, I'm on there as well. Under Ed, so you can find me. That's it.
Hi guys, so I'm Ali from HackScience, and I'm just going to set the scene a bit for disrupting the lab, as well as tell you guys a bit about what we're doing at HackScience. So we all know that scientific research changes the world, from breakthrough technologies to life-saving medicines. But research is slow and it's becoming increasingly more and more expensive. So at HackScience, we accelerate research by automating manual lab processes, and we do this by using IoT and web technologies. And this is our team. There's 5 of us right now, and we're growing, and most of us went to Imperial. And we bring together deep expertise in hardware, software, product, and science. I myself am a PhD student, and I previously founded a hardware startup. I'm here tonight. Ignacio's here, and also Henry, so come talk to one of us. So as an example of a researcher, we're going to talk about one of our friends at Imperial. This is Dr. Michael Delves, and he's a fantastic malaria researcher. Some of you guys might actually know him. And he's got great ideas about stuff to research, but unfortunately Michael has a problem that stops him in his tracks. And that is that he has to spend 3 hours a day while his malaria cultures are growing, manually replenishing the growth media. So that looks like this. He will take each flask out of the incubator, suck out the old media by hand, and replace it by hand similarly. And Michael's actually not alone. We've spoken to researchers at some of the top institutions in the world, including UCL, KCL, Imperial, even Stanford in the States. And what we found is that 62% of researchers are spending more than half their time in the lab doing things by hand, which is a crazy amount of time. Now that stat alone should tell you guys that the lab needs disrupting. So you think there must be a better way. Well, in Michael's case, there is another option, but it costs £250,000. So this is like an automated cell cultivating machine. And this is a trend we see across not just automated tools in science today, but generally scientific equipment. It's super expensive. Often it takes up a lot of space, and it's just not that easy to use. I mean, you guys have probably all used the piece of science equipment connected to the Windows 98 computer, so I don't need to talk about that too much. But there are new options appearing. So this is a new option that could be available to Michael. He could outsource some of his research to a cloud lab. So for those of you that don't know, a cloud lab is essentially a laboratory in a warehouse somewhere in the world, and you send them your protocols, they run it for you, and they send you back the data. This is pretty interesting. But there are also other options appearing now, and that's thanks to the maker movement. So the maker movement is essentially a movement of people making things for themselves, one-off things, or things in small quantities. And the reason that's become viable and possible is because it's super easy to create software-controlled electronics now. Sensors have become super cheap because of mobile phones, and you can print things of any shape quickly and easily using a 3D printer. And this has actually led to cheaper versions of scientific equipment starting to be made. This is leading to a citizen science revolution where we're seeing people who couldn't traditionally afford to have scientific equipment buy equipment and run their own experiments outside of a lab environment. This is actually a super early prototype of what Bethan will be speaking about later, just an example. But at HackScience, we're interested to see whether we could use that same maker technology to automate some of those manual lab processes I mentioned. So to test this idea out, earlier in the year, we brought together scientists and engineers at Imperial, and we ran the world's first lab automation hackathon. And it went a bit like this. So first, the researchers, we had an ideation session where the researchers pitched their manual lab processes and formed teams with engineers. Next, we trained everybody in Arduino, 3D printing, laser cutting. And finally, we had the 2-day hackathon, which is essentially 2 days of intense building, going from the initial idea to working prototypes. But at the end of those 2 days, we were blown away because people had proved that affordable automation was possible. And they proved it across 9 different areas of research: solar, malaria, cancer, just to name 3 of them. And just to give you an example of some of those tools, so here are some of the tools that were created. I'm going to dive in on 3 of those just to give you an example of what's possible. So one team created a wireless cell density sensor. You drop this in your flask of cells, and essentially it wirelessly takes optical density measurements, or OD600 measurements, and that lets you know how many cells you have per unit volume. It's pretty cool. One team built a Coulter counter, so that lets you count cells passing a particular point in a microfluidic channel. Typically, these cost hundreds of thousands of pounds. These guys built it for 400, which is pretty great. And finally, someone automated column chromatography. So if there are any chemists out there, this is super laborious normally. These guys automated it just with some cheap optical sensors and some clever mechanical parts, which is very cool. And you remember Michael? Well, we're actually working with Michael to automate his process. So this is an early prototype of our cell cultivating tool. It's not going to cost £250,000. It's going to be very cheap. And yeah, we've actually progressed past that now and created something that can clip straight onto these flasks that people cultivating, so that's pretty cool. And yeah, if you guys are interested in that, so I'm sure there's some of you in the crowd who grow cells. If you're interested in this, just take a photo of this link, sign up. And if you use the referral code SD for Science Disrupts, we can add you guys to the top of the list for like user testing. We're just like testing out the tool right now. So an interesting thing we've seen in the software world is open source. This has allowed software projects to basically have massive distributed teams across the world working on a project 24 hours a day, 7 days a week, and combining all their ideas. And we want to use some of those same forces in science. So we're planning on open sourcing all of our own tools and also all the tools from the hackathon. And we have a vision for our platform to kind of have millions of open tools. We want the sharing of designs for open tools to replace the sharing of methods between scientists. And that's going to make reproducibility just so easy. So that's our kind of vision. And obviously, researchers deserve the credit for the work they do, so we're working on making it super easy to cite and keep track of citations. And the final thing we noticed from the hackathon and we're working on is that people could build automated tools, but when it came to building that next level of software to allow you to control and monitor your tool remotely, that's where people really struggled, and that's much more the realm of a software engineer. But we wanted to make that easier too. So we've created the HackScience Hub. Essentially, you plug your new tool into one of these 4 USB ports, and you can create a cloud-based interface for your tool using a drag-and-drop interface. It's kind of like LabVIEW in the cloud, but easier, and you don't need to know anything about how the internet works, which is pretty cool. So yeah, that's coming mid-2017, and again, if any of you guys are makers and want to try that out, we're really happy to get involved. We've actually used it to build an interface for our own cell culturing tool. This took us about an hour to put together, but I think it's already better than a lot of the interfaces we see for scientific tools today. So yeah, that's a little bit about what we do at HackScience. And if you guys are interested in knowing about our next hackathon or learning about any automated tools that might come out, if you go to this link, bit.ly/hsmail, you can sign up and we'll let you know about those things. And yeah, just get in touch. We love hearing from you guys. We love chatting to researchers about what you guys do, anyone who wants anything automated, anyone who's making stuff, and yeah, just come talk to us. Cool.
I feel like I have big shoes to follow, given that we have an audience of people who are coming here from like lab backgrounds. So I want to start off where I was 4 years ago. 4 years ago, I was an undergrad. I was doing a project called iGEM, which is a summer biology project. You build a genetic circuit over the course of a summer. Now, we heard about this group called the London Biohackspace. They belong to this growing movement of people that do molecular biology outside the lab called DIY Bio. And being clever students, we thought it would be really awesome if we went to this space and they could help us with our lab work and, we'd win a prize in the competition. It turned out that this particular group of people were actually mostly computer scientists and software developers that were really fascinated with the idea of biology as a technology, and you can learn to use it in the same way that you could learn coding. So they grabbed us and they got us to teach them molecular biology instead for the summer. But along the way, we got to do some really interesting projects, which follows a little bit on from HackScience, where so here we're building a shaker incubator within about 2 hours, and we're doing that with bits of wire and bits of wood that we hacked together and sellotaped tubes into this fridge box, and it lasted just long enough, 24 hours, to grow up our cells so that we could make them competent for transformation. So that was really a kind of an eye-opening experience to see that you could do lab work and molecular biology outside of a traditional setting. And it also got me and my co-founder Philip totally addicted. We finished the summer project, but we wanted to carry on. We couldn't believe that we had to wait until a PhD or for a summer internship to carry on doing genetics and molecular biology and this amazing thing called synthetic biology, despite however many failures you might have in the lab. So we kind of stayed involved. We started to go along to these European meetups for DIY bio, and the discussions all revolved around how do you bring more people in? How do you take someone who has no experience of pipetting? They have no concept of, is DNA bigger than protein? What context do they have to understand those kind of questions? How do you take someone like that and bring them in and hold their hand so that they can then go on to build their own genetic circuit? Or even at a simple level, say, I want to test this plant to see if it's genetically modified. So our concept, Philip and I, coming out of that was this idea of, okay, well, look at the personal computer revolution. That's something that, that gets brought up a lot in synthetic biology particularly. If you make the tools smaller and more accessible, what can you do with them? Who starts to use them? So I mean there you have computers, they're massive, people work inside them, they're only available to industry and academia really, and then you make them smaller, and it's not just that they're smaller and they're cute, it's also that the applications and the people sat behind the computer starts changing. So we're taking a bit of a gamble. We're saying, okay, let's take some of the most interesting bits of lab equipment, not particularly like revolutionary, but the most useful ones, and let's make them tiny and small and cute, and let's see who the users are and how the users change. So at the moment, our kind of vision and our kind of pitch is we want to build this ecosystem that anyone, your cousin, potentially your mother, a farmer, could use to come and learn about molecular biology and DNA and actually start experimenting and start taking samples from plants, from their own saliva, and get results. So our first step towards that is this biology lab, and like I said, it's not particularly anything sexy or innovative, it's just we're making it for someone different, and we're saying this is who it's for and you're able to use it. And the focus there is really on the user design. The user experience. So the idea is that we're boiling it down to trying to make it really simple and saying step by step. Let's not use jargon like resuspension of cells. Let's focus on saying, okay, step 1, step 2, step 3. How do you try and automate this, but in a human fashion? So this idea kind of came out about 3 years ago. We started, as Ali showed you, we started with this Darwin toolbox, this lab in a box. And it progressed from there, and it became our beta version, which is BentoLab, which we then actually went on to send to people around the world. So we have Alex, who is wacky and travels all kind of countries doing different things, but one of the things he was doing was looking at Zika virus in Brazil, and trying to do kind of on-site pre-diagnostics. And then we have Gianpaolo, who runs an awesome project called Beer Decoded. He takes the yeast from different beers, and then he looks at the different genetic sequences to try and relate them. So he can say, okay, this Red Church IPA, it's pretty connected to this tiny Rebel IPA, and you're probably going to like it if that's what you like. And then you have David. And David is one of my favorite beta testers because he's a biohacker and he doesn't even know that that's what he is. So David collects mushrooms, and he's on the hunt for like this particular black cap species. And how he wants to do that is by identifying it with DNA. And so originally he was working with the university, and now he's trying to do it himself at home in a garage. And we're helping him do that. And actually, we've started working with Kew Gardens to help them get more people doing exactly that. And then, of course, you have education, and you have people that are bringing the lab with them into like school classes and just making that a lot more hands-on. Our aim with the lab was to take it out of the thousands of pounds and into the hundreds, and hopefully getting it to the point where actually that equipment, it's minimal, like Ali was saying, it's kind of the equipment is not the barrier. The barrier is the knowledge, the barrier is the consumables and getting started. It's not getting a hold of the equipment. So following on from our beta testing, we went on and we built 20 of them by hand. And I did the soldering, and it was a very, very painful process, particularly because I'm a biochemist. And we took all of that feedback and we put it into building a new version. And actually, that's something that we crowdfunded earlier this year in April. And we had about 200 people go ahead and get this lab. And as with all kinds of projects like this, it's ended up taking a lot longer than we anticipated. But we'll be shipping out from January. So we're pretty excited because we've got these 200 people, 100 of which are researchers or more about, they know what they're doing, and then 100 of them have absolutely no idea, and that's really exciting. Although the responsibility lies on me because I'm the one designing all the protocols and the starter kit and stuff. So as I said, I think initially when we got started, we were thinking about, okay, make the hardware smaller and cheaper and more user-friendly. But actually, it's not just about that. It's going beyond the equipment and thinking, how do you bring someone into the ecosystem and what other support do they need? And actually, for us, the goal is thinking to, okay, well, you can learn how to code within a couple of months. You can build an app, and you can relatively easily distribute that online, and other people, if they're interested, they'll buy and use it. So we had all these questions out of the Kickstarter, people who were beekeepers, and they were emailing us saying, how do I know if my bees are African bees? Or I'm a hops farmer, and I sell this to microbreweries. How do I know if my hops are wild or if they're the ones that I started off with? So for us now the question is, okay, well, what if we can take one person like that who's asking a really interesting question, and we can help them to develop a kit or an application around that, and then they can go off and they can meet other people via our community, because I know one mushroom guy, I know 100 mushroom guys, and I'm not joking, there are 100, much more actually, mushroom guys out there. And they're all interested in these types of questions, but it's having that support to get the answer to that question that they're starting off with. So I'm not sure how I'm doing for 15 minutes, just because I've been doing a lot of different. I'm good. So this is my why is that important slide. This was a guy that we met at Shenzhen Maker Fair. He's about 3, and he spent, like, not joking, 45 minutes with us just pipetting water in and out. And his parents actually left him with us, and then they came back to pick him up. So if I wanted to, looking at DIY bio and biohacking and hacking equipment, I could go onto eBay and I can buy enough equipment to do a PCR test and a gel diagnostic test for maybe about £1,000 or even less if I use all my lab friends to get me free equipment. And with that, you can do all kinds of interesting things. You can look at your own DNA to see if you have the SNP that means that you're more likely to detect bitterness, or that you're likely to be socially irritable, or whatever those kind of science papers are coming out and saying. And you can engage with that research and give it a go yourself. Or you can start to use it for more serious things, like if you wanted to, looking at whether or not you have mutations that are linked to Alzheimer's or cancer. And these are tests that people are already doing every day. They're doing it with 23andMe, they're doing it, and they're getting the results back, and they don't totally understand what those results mean. Partly because as a society and as a culture, we don't engage a lot with genetics and molecular biology if we're not outside of that field. And that's exactly what we want to do. We want to make something that's incredibly fun, that brings people in because they're asking their own questions, and they get hands-on with genetics and biotech. And ideally, that someone like this, in 10, 15 years, they're going to be the next computer coder in their bedroom at 15, making baby unicorns or whatever. And that they're going to be bioliterate. Cool. So that's me done. Oh, I'm not going to...
Okay, cool. So I also have very big steps, or very big shoes to fill. I don't know very many, probably fewer than one mushroom folks, but I'll do my best. So I'm going to tell you a little bit about the making lab that I'm setting up at the Francis Crick Institute, which is just about 10 minutes that way across St. Pancras Station. And as you can see, it's a building site right now. The lab is due to open soon, but there are a few things that need to be figured out first. And so I've actually titled this talk, Making the Making Lab. And I'll tell you a little bit more conceptually about what we're going to be doing and how we aim to change the way that science is done, both for us and folks working around us and with us. So the Making Lab is, we try to focus on these several key areas, and I'm going to touch on a few of them, not all of them, so I won't bore you. First, a little bit about the Crick. They say that their first strategic priority is to pursue discovery without boundaries. And what I think that means is bridging the physical and life sciences to do things that aren't commonly done in life sciences. But what they have given me a little bit of a hard time with is erecting a wall that borders my lab. And that yellow arrow is pointing to that wall. And believe it or not, that wall had to be shipped from Spain, and the shipment got delayed, and that's delayed the lab opening. So despite the fact that the Crick is telling us that they're pursuing discovery without boundaries, they're not giving me the wall or the boundary to do it. Anyway, so I think the really key concept that the Making Lab tries to get at is that we want to be imaginative. The complexity of biological problems today and the intricacies oftentimes demands an interdisciplinary approach between life scientists and physical scientists that requires a lot of failure and novel and imaginative solutions. And those things are not easily pursued in traditional labs. And so we try to create a space where people can co-work and collaborate and fail together and succeed together. And we are aiming to foster that spirit through being a few things that I'm going to elaborate on a little bit: bold, open, collaborative, and communicative. So first, we're trying to be bold, and this will give you a little bit of an idea of what we actually do. So first, we focus on microfabrication, so things like photolithography to generate microfluidic devices and micropattern devices. A little bit of macrofabrication, so 3D printing, CNC milling, a little bit of electronic fabrication, PCB, manufacture and developing sensors and actuators, and all of those areas of fabrication overlap and allow us to create some pretty cool devices. We are trying to be accessible, and that means a few things. First, there are some companies that exist to do some of what we do. They have some drawbacks. None of those companies are embedded in the Francis Crick Institute like our lab is. And oftentimes, as most of you know, or all of you know, solutions to scientific problems require multiple steps. And the company that can provide a solution to step 1 is usually not the same company that can provide a solution to step 2. It's not the same company that can provide a solution to step 3, et cetera, et cetera. And that ends up costing you as researchers a lot of time and money, and we aim to overcome that. Secondly, the traditional areas where this kind of physical science and life sciences has been done require either a clean room, as indicated in the picture on the left, or in this kind of very stereotypical image of a molecular biologist, unlike what we just saw. And neither of those venues are very conducive to communication or collaboration. And so the physical space of our Making Lab is diagrammed here, and don't worry about reading any details, but just suffice it to say that it's a space that contains benches where physical scientists and life scientists can work together in the same area. And as you guys know, the refinements that oftentimes underlie scientific successes are out of reach in the traditional settings, and we're aiming to kind of expedite the process, make it more efficient and more cost efficient from a cost and time standpoint, and allow people to work together. We're aiming to be collaborative, and that means that first, we don't need that much equipment or resources to make the stuff that we're going to make, and there are a finite number of things that we need to possess, and we aim to teach individual researchers and train them on the pieces of equipment that do exist. And in this cartoon diagram of people, notice that they're arranged horizontally, and that was very specific. We aim not to create a vertical environment where there is, say, a director of the lab and then people underneath that director, but more of a horizontal environment where one should feel free to talk to anyone else and share questions, curiosities, failures, successes, etc. Part of our rationale for doing this is that there's a lot of experience and expertise amongst other Crick core research groups that we aim to take advantage of in the first instance. And finally, we're collaborative not just within the Crick or within London, but internationally as well. There's a wonderful new microfluidics institute in Paris called IPGG, and we collaborate a lot with them. And I might say that we've hosted at UCL, where I'm currently doing my postdoc, these biological making workshops that have been pretty successful in 2015, 2016. I know that a few of you came to those. The next edition might be at the Crick, it might be at IPGG. If it's in London, I encourage all you guys to come. I think it's a really fun time. And finally, we aim to be communicative. I think we all in this audience believe that the impact of any scientific research is limited by our ability to communicate it to an external audience, be that an external audience of experts or an external audience of non-experts. And that's part of the reason why I'm really happy to be here talking to you guys today. And luckily, the Francis Crick Institute and other affiliations that I have both external and internal mechanisms to disseminate the information that we're going to learn. So I hope I've given you a little bit of a sketch about what the Making Lab is going to be. And I might give you one application. And this was a really fun project that we started when I was a postdoc in Dresden, Germany. And Martin, who's the lead author on this paper that came out last year, and we did this funny thing about putting cells in microfluidic channels. So basically, we put individual cells in miniature plumbing. And we didn't think much interesting would happen, but we were really surprised when the cells started moving. We're like, well, what's going on? And it turns out that when we made the plumbing a little bit bigger, the cells slowed down, and we made it a little bit smaller, the cells sped up. That was a little bit puzzling too. And we tried to come up with explanations. We failed with a lot of those explanations, but we finally hypothesized that maybe a cell's ability to move has something to do with the friction that it can exert on its environment. And so we created, designed and manufactured this microfluidic device, which is kind of schematized here, that allowed us to measure the friction that cells exert with their surfaces, with their surrounding environment directly. And the results of those friction measurements were consistent. That is, the higher the friction, the more efficiently the cell could move. And I wish I could have embedded the videos in this presentation. Sadly, I couldn't, but I encourage you, if you're interested, to read the paper and dig into supplementary material a little bit where the videos actually are. So that's what I wanted to tell you guys today, and I hope that's given you a little bit of a sketch about what the Making Lab is envisioned to be, as well as one example of an application. And by all means, if anything is unclear, feel free to talk to me more in the panel or afterwards, just outside. Thanks.
Protovision is a lab tool designed to make running your experiments and day-to-day life in the lab easier. Built around augmented, video-enabled glasses, the system is designed to help you to run your experiments, connecting you directly to your data, your team, and even your collaborators outside the lab, no matter where in the world they are. Simply put the glasses on and they activate automatically. Operated by voice or gesture, you can access your email, schedule, all previous results as well as any current protocols you're running. The system augments the lab with live data and information, which can be easily seen and accessed by simple icons that attach themselves to equipment. Once ready, the day's protocols can be accessed and progress reviewed. Results can be reviewed or compared to results from previous experiments. Results can be shared with your PI or other members of the team immediately. Video conferencing then allows you to discuss results without leaving your bench. With the use of custom tools, the system can help make experiments easier to manage. Using inbuilt image tracking, the system can help in tasks such as counting fruit flies, saving time and avoiding miscounts. You can also keep track of materials, log fridge inventory, and reorder supplies. Research notes and videos captured during the day can then be shared and stored wirelessly to any repository. Content can then be accessed anytime and by any connected device to be shared with collaborators.
So that is ProtoVision. And as I say, we created that 3 years ago thinking this could be reality by now. 3 years ago, if you asked me about the next company, I was thinking, okay, maybe we're 5, 10 years away from that, which is one of the things about technology is you can never predict. The pace at which we're seeing change in what people like Ali are doing and the other speakers, in the lab environment is phenomenal, one of our jobs is to try and keep track of that and keep pace with that. But it's more than a one-man job, that's for sure. So without further ado, let me play the next video from Transcriptic. So that's right.
When we think about the great biotech companies, their value is as enterprises is really in the scientific knowledge and scientific content there. They're really information companies. But that's very different from how we usually think about them. The mission behind Transcriptic is to be the world's lab. We see the world in terms of devices. This way of thinking about it is really different.
In order to make research much more efficient, we really want to drop the hands-on experimental time down to almost zero, or just a few clicks on our website. So in order to do that, we've built a platform that will automate the entire workflow for a molecular biology experiment.
There's a lot of things a lab tech does in the Performance protocol that are based on intuition or experience, and we include all of that in our software.
All these little details that are extraordinarily important how you map things onto the robotics.
So no longer do you need to have technicians doing a lot of manual pipetting or moving plates between pieces of equipment, but be able to basically input your sample, create an order on the website, click go, and it will autonomously go through the entire workflow for you. So you can spend that extra time doing more science.
The science team brings together the products of the hardware and software teams to develop the protocols used by our customers. One of the greatest assets of the company is the science team has experience in both biology and software development.
We do everything from building our own software interfaces all the way down to nuts and bolts of hardware. This leads to lower costs, total customization, and a more wholesome design.
We have to think very hard about how do we provide that high level of fidelity to the customer. If this was just a system that was useful for running the same thing 10,000 times, that would be a much more limited usefulness. But I think the heart of this is that we can not just run one thing 1,000 times, like the industry does today with these robotics, but we can also do 1,000 things once. And that really opens up how people think about robotics. Doing life science research shouldn't require millions of dollars in lab equipment or hours at a bench. This is something you should be able to do wherever you are through a laptop.